Welcome back. Now that we've seen the differences between muscle cells, between our cardiac muscle, our heart, our skeletal muscle, and the smooth muscle, now we need to take a little bit of a look at how they're actually controlled and a little bit at the differences in their physiology, not just what they look like. So when we look at the actual muscle cells themselves, we see the differences that we saw in terms of the anatomy, their changes in the shape of the cells, going from cylindrical, long cylinders to branching, having intercalated discs to being spindle shaped as far as the cells go. All of those changes also lead to changes in terms of how they function within the different types that are there as well. And so regardless of whether we're talking about skeletal, cardiac, or smooth muscle, they all have some things in common. And so the first is the proteins that are involved. And so we have our contractile proteins, our actin and our myosin. Those are involved in all three types of cells that are there. We're going to control, change a little bit of the regulatory proteins in smooth muscle, but that's really going to be it. The actual contractile proteins are there. They're the same. We utilize them for all three different types. They all require ATP. We've got to have energy. And so they're going to need energy. They're going to need oxygen. They're going to need a supply of glucose. They need to get that ATP into the muscles themselves. Depending upon the muscle type, their requirements are going to vary. A smooth muscle is going to require a lot less than, say, the cardiac muscle. The cardiac muscle is generally going to need more than most skeletal muscles most of the time. But at times, skeletal muscle is going to need more there as well. The other thing that kind of binds them all together is calcium. And so calcium inside the cell is what's going to be essentially our trigger for muscle contraction. So no matter who we're looking at, no matter what type of muscle we're looking at, they're all going to require a calcium trigger or a calcium key to unlock the ability for our myosin to attach to our actin on the cells themselves. So these are kind of the similarities between the three. And when it comes to control of the muscles, this is where we start to diverge quite a bit. And so, as we've said before, skeletal muscle is voluntary. So we can control when it's contracting and when it's relaxing. This control comes from the nervous system, and it more specifically comes from our somatic nervous system. So we have motor nerve pathways, we have descending pathways that are in control here. And this is going to go out and it's going to control skeletal muscle. The type of control is what we call neurogenic. And so the actual contraction themselves are initiated by the nervous system. And so although it is voluntary, we control when we want it to contract, when it's not, the actual process itself of stimulating the muscle fiber to contract is done so by a neurogenic process. And so the nervous system is telling it when to contract and when to relax. If we've seen before where if we take away that nervous input to the muscle starts to atrophy. It needs to have that kind of constant little stimulation all the time. And if we take it away, then that muscle no longer is going to contract. It's no longer going to have that ability to have a normal contraction that's there. So we rely upon a neurogenic process in order to initiate the actual contraction of the muscles themselves. Cardiac and smooth muscle are autonomic, and so these are involuntary. We don't have any control over when they contract and when they relax. The autonomic side of the nervous system, remember, has two different portions. We have a sympathetic and a parasympathetic pathway. The sympathetic being that of our fight or flight, and our parasympathetics being our rest and digest. So we have the ability to kind of turn things up or turn things down. They're always working in opposite directions of one another in general. And so when we're looking at these muscles, we're either going to be turning things on, turning up the processes, or we're going to be turning them down depending upon which side of that autonomic system is predominant for that particular muscle type. For cardiac muscle, not only does it have influence from the autonomics, but it also has a myogenic type of control. In other words, the actual stimulus for muscle contraction is coming from the muscle cells themselves. It's coming from the muscle. And so it's initiated from the actual cardiac cells themselves. We talked before that we had pacemaker cells. And so those special pacemaker cells that are inside the muscle cell generate the input that's there. And then the nervous system, either our parasympathetic nervous system or our sympathetic nervous system, is then going to modify the contraction. And so it's going to either increase heart rate, increase the strength of the contraction, or it's going to decrease them. Decrease our heart rate, decrease our strength of the contraction itself. And so we have the opposite opposing of our sympathetics and our parasympathetics with it. But the ultimate starter for this process is cardiac muscle. Now, in the past, we used to do experimentation with live organisms. And so we used to have the process where we could utilize animals in the labs themselves. And so for cardiac muscle, we would use things like turtles. And so turtles have the ability to, once they've been euthanized, we can open up the shell and look at their heart. And you'll see that the heart keeps on contracting. And it's contracting for a very long period of time. And the heart is initiating on itself. The nervous system has been disrupted. There's no nervous input into the heart. And then we can look and see that it continues to contract. This process itself was kind of memorialized or sensationalized in the Indiana Jones movie, in which the high priest removes the heart from one of the people out of the chest, and it continues to beat inside his hand. And in reality, that really can happen. If we take away that nervous input, the heart will still contract. This is why someone can be brain dead. They can have no higher brain function, but the heart still continues to contract. It's still doing its job because it has that myogenic control for the system itself. And so it has that ability to contract all on its own. Smooth muscle, on the other hand, has both myogenic and neurogenic control. So here we have both types of control for the smooth muscle. And this is another one that we used to do in the lab as well. For the myogenic side is that we would have different types of muscle available. And for the intestines, the small intestines has a great deal of smooth muscle in it, and we'd be able to section small pieces of smooth muscle from either rats or frogs, and students would then be able to go and get a small piece, and they would see that it's kind of wiggling around. It would be almost look like a worm kind of contortioning back and forth, and that was that myogenic control of the muscle. That muscle was being controlled entirely by the cells inside the muscle itself. Again, the nervous system does have that ability to modify it. And so different areas of the smooth muscle in terms of location of smooth muscle, type of smooth muscle, have either myogenic or neurogenic control, depending upon where they are. If they do have myogenic control, they still have that same as the cardiac muscle, they still have modification available through the autonomic nervous system. And so we're either going to have sympathetics or parasympathetics changing things. So parasympathetics, rest and digest, digestive system, that's going to increase that activity within the smooth muscle themselves. That's there. And so we can add things into the smooth muscle and see them increase or decrease their contraction based upon the chemical that is given to them. On the neurogenic side, this is where we have nervous system involved. This is either the sympathetics or parasympathetics. In some places, it's all sympathetics for the muscles themselves. So in places like many of our blood vessels, we have only sympathetic control. We just change the amount of control that's there. But in most of them, they do have, again, a parasympathetic and a sympathetic input. And that's going to initiate the actual contraction of the muscles themselves. They don't have the ability to do it all on their own. That propagation of the action potential is made possible in both cardiac muscle and smooth muscle via gap junctions. So cardiac muscle has very little sarcoplasmic reticulum. Smooth muscle basically has no sarcoplasmic reticulum. So we've got to be able to get the calcium ions, sodium ions through the cells through these gap junctions. And so the gap junction itself is going to allow for these small ions to make their way through, and they're able to move from one cell to the next. And when it comes to things like peristalsis, we can literally see where there's, in a sense, kind of like a wave of contraction and relaxation along the muscle itself. It's allowing for that contraction and relaxation to occur all throughout the pathway of the muscles that are in there. And similar to that of the cardiac muscle, if we watched a heart contracting, especially if it's slowly contracting, it would almost look like somebody was squeezing the heart from one end to the other. So the apex of the heart squeezing up towards the base of the heart as it contracts. And that's that wave of contraction that's occurring across the cells themselves. And it makes it jump from one cell to the next cell to the next cell, all along the pathway of those muscle cells. And so we're able to connect one cell to the next and get over the fact that they don't have the ability to very quickly dump all of that calcium into the inside of the cell. So not only are there myogenic potential controls of the muscle jumping from one cell to the next, so is that action potential as it moves along through the muscles.